we're investigating how pH affects the rate of amylase activity.
Okay, Section 1, Key Enzyme Ideas. Before we even think about touching any equipment, we need to understand the science behind what we're actually doing. Why does this experiment even work?
So, what on earth is an enzyme? Well, at its core, it's a biological catalyst. It's basically a special type of protein that's like a little factory worker with one very specific job: to speed up a chemical reaction. Like, massively speed it up. And the cool thing is, it does its job without ever getting used up itself. And the best way to get your head around this is the lock and key theory. It's a brilliant analogy. The enzyme is the lock, and the molecule it's working on, the substrate, which in our case is starch, is the key. It's a perfect fit. Only one specific key can open that lock. And this leads us to the most important bit of that lock, the active site. This is the uniquely shaped region where the key, our substrate, actually fits in and where all the action happens. That shape, it's everything. It's absolutely crucial. So this is where pH comes in. At its perfect, optimum pH, the active site is exactly the right shape and the reaction is flying along at top speed. But if the pH gets too high or too low, it's bad news. The bonds that hold the enzyme together start to break, which completely changes the shape of the active site. The key just doesn't fit anymore. And when that happens, we have a special word for it. The enzyme is denatured. It's game over for that reaction.
Right, that's the theory. Let's move on to Section 2, the practical method. Time to see how this all plays out in the lab with a step-by-step guide.
Okay, first up, let's get our equipment sorted. You're going to need your amylase and your starch solution, obviously. But the really important bit here, the buffer solutions. Now, what's a buffer? It's basically a clever liquid that keeps the pH steady. So if we use a pH 5 buffer, we know our reaction will stay at pH 5. It's how we control our variable. And of course, we'll need iodine, which is our trusty indicator for starch. So the method itself is what we call a time series experiment. First, get your spotting tile ready with a drop of iodine in each well. Super important, get all your solutions to the right temperature using a water bath. Then, in a test tube, you'll mix your amylase with one of your pH buffers. And here's the T moment. Add the starch and hit that stop clock immediately. Then, every 30 seconds, you take a tiny drop of the mixture and pop it into an iodine well. You keep doing this until the iodine stops turning that familiar blue-black and just stays, well, its normal brown-orange color. That's your signal. All the starch has been broken down. Stop the clock and write down that time.
Okay, you've done the experiment. Great job. Now for Section 3, Results and Analysis. This is where we turn those times you recorded into some meaningful data.
You've absolutely got to put your results into a clear, organized table. Something like this is perfect. You'll have a column for your independent variable, that's the thing you changed, so the pH. Then a column for your dependent variable, the thing you measured, so the time it took. But hang on, what's that third column? The rate of reaction? Yeah, we're going to have to calculate.
So why can't we just use the time? Well, to make our results easy to compare, we calculate the rate of reaction. The formula couldn't be simpler. It's just rate equals 1 divided by time. And it makes perfect sense, right? If a reaction is really fast, the time is small. And 1 divided by a small number gives you a big number. So a short time equals a high rate. Easy.
Now that you've got your rates for each pH, it's time to visualize that data. You're going to plot a graph. The rate of reaction goes up the side, on the y-axis, and the pH goes along the bottom, on the x-axis. And when you plot your points, a really clear pattern should just jump out at you. You'll see the reaction is fastest at one particular pH.
And that peak on your graph, that's the money shot. That's the enzyme's optimum pH. It's the point where it's working at its absolute best.
Section 4, Evaluation and Exam Skills, is all about how you secure those top marks.
Look, if you want your results to be valid, you have to run a fair test. That means you only change one thing, in this case, the pH. Everything else has to stay exactly the same. We call these our control variables. And temperature is a massive one here, because it also affects how fast enzymes work. That's why we use that water bath. You also have to be really careful to use the same concentration and the same volume of your solutions every single time.
You can bet an examiner is going to ask about potential errors, and a classic one for this practical is judging that color change. Is it still a little bit blue or is it properly brown-orange? It can be tricky and a bit subjective. Of course, you could also have things like a slight delay in starting the clock or being a bit off with your measurements. The best way to make your results more reliable, repeat the experiment at least 3 times for each pH and calculate a mean. That also makes it super obvious if you've got a weird, anomalous result.
Okay, time to put this into practice. Here's a typical exam question you might get. Explain why the rate of reaction decreases at pH values above the optimum. It's worth 4 marks. So, pause for a second. How would you structure your answer to make sure you bag every single one of those marks?
Right, here's how you do it. You need a logical chain of events. First mark, say that pH is above the optimum, start to break the bonds inside the enzyme. Second mark, explain that this changes the specific 3D shape of the active site. Third mark, this is the big one, you've got to use the keyword, the enzyme is denatured. And for your fourth and final mark, you link it all together. The substrate can't fit into the active site anymore. So you get fewer successful collisions. Boom, 4 marks.
So let's do a super quick final recap to make sure all the key bits have stuck.
Okay, here are the absolute need-to-knows. 1. Enzymes have an optimum pH where they work fastest. 2. Extreme pH levels will denature the enzyme. 3. You calculate the rate using that simple formula. Rate equals 1 over time. 4. Your graph should have a clear peak at that optimum pH. And 5. To make it a fair test, you have to control your temperature, concentrations, and volumes. Got it?
And I'll leave you with this to think about. We've seen that amylase likes a nice neutral pH 7. So why do enzymes in your stomach, which is full of acid, work best at a crazy low pH of 2? And then, just a bit further down in your small intestine, other enzymes work best at an alkaline pH of 8. What does that tell you about how amazing and specific these little biological machines really are?